Dear colleagues, we will start our first session, and it's about cancer genetics and precision medicine. Dear colleagues, we will start our first session about cancer genetics and precision medicine. And just before we start, I would like to mention some housekeeping rules. After each lecture, it will be time for 1 to 3 questions, and if you are in person here, you will come to the microphones in the hall. If you are taking part virtually, please write the questions in the chat and we will deliver to our lecturers. And the first speaker of this session is Professor Pieters, Chief of Medical Officer at Princess Máxima Center of Pediatric Oncology and Professor of Pediatric Oncology in the University of Utrecht. The floor is yours, please.
Okay, thank you. Thanks for the invitation to speak here. I didn't know that I was an expert in precision medicine, but from the booklet I learned that I was. So I hope I will not disappoint you with my speech. I was asked by Elina first to start and tell you something about the Princess Máxima Center and how we came to this National Childhood Cancer Center. So if you don't want to hear this, skip the first 5 slides and then you can go on to the precision medicine. But we have in the Netherlands, we have 17 million people living there, and there are about 600 children diagnosed with cancer each year. And these children were treated in 7 different pediatric oncology centers. And as you probably know, every type of childhood cancer is rare. And if you see 7 patients a year in the Netherlands with an hepatoblastoma, so liver cancer, it doesn't seem very smart to do this on 7 places. So what we did was we gave up these 7 centers and we started a completely new National Childhood Cancer Center in Utrecht. So all the old centers disappeared and we merged all the care and research in one National Childhood Cancer Center. And this was done by the Dutch Childhood Oncology Group, who is the organization in which all professionals are organized, together with the parents' organization. And these 2 organizations started a legal entity and they own this center. So it's owned by the patients and by the professionals. So in 2007 we had the first ideas to do this, and in 2014 we started on a small scale by merging all the children with solid tumors, and in 2016 with merging all the research, and we rented several floors for this in the University Medical Center of Utrecht, and in the meantime we were building our new building, which was ready in 2018, and then from one day to another all the patients and professionals from these other 7 institutes moved to Utrecht at the same day, actually on the same morning. And this looks complex, but it's very simple. You have to pick up your things and drive to another center and simply go on with what you did before. So this is our Princess Máxima Center. It is situated here, and this is an enlarged part of the building. The last part is the research part, and this is the care part. And the Princess Máxima Center is situated on the Utrecht Science Park campus. We are physically connected via bridge to the Wilhelmina Children's Hospital, and we use their intensive care unit and their surgical rooms, but we have our own surgeons, our own neurosurgeons, but we use their operation theaters. And this is the Hospital for Adults, University Medical Center of Utrecht, and this is the campus with all the faculties for biology, psychology, pharmacy, etc. So we have a lot of possibilities to collaborate. Well, having established this Princess Máxima Center in 2018, I will give you an idea about what this center is. So it's a comprehensive childhood cancer center. So we only treat children with cancer, no other patients. We only do research on childhood cancer. As a kind of side effect of this merge, we became the largest childhood cancer center in Europe. Well, being big itself doesn't mean anything, but it will attract good researchers and good clinicians that want to work for our organization and for our children, and therefore many people came to work for us. So we see roughly 600 newly diagnosed children and 150 children with a relapsed form of cancer, and we have roughly 1,500 people working in our center, of which 900 are clinical people and 450 in the research. Because many people come from different countries, we have 40 different nationalities working at our center, and we have 36 research groups, and the hospital is built that connected to each patient room, there is a parent room, so that parents can be close to their child, and we have 87 of these units, and we have 35 daycare beds that we use twice a day, and we have a budget of 230 million annually. So the scientific output, so if you merge all these research, you can become very productive. And as an example, in 2021, we published more than 600 international papers. And we do a lot of trials. And this will also be the step to the precision medicine program. So we have a trial and data center in which 100 people work, and we run now at this moment more than 130 clinical studies. And one of the aspects of our center is also a very big long-term follow-up program, which goes into adulthood and in which we see more than 15,000 long-term survivors of childhood cancer. And of course of this, because all patients are in the same institute, it's easy to get access to the material of the patient. This sounds a little ugly, but the biopsies that are done are available also for the research and not only for the diagnostics. So I switched to the Pediatric Precision Oncology program, and this is called in our center the ITER program, Individualized Therapies for Children with Cancer. And if you stop listening to me, that's okay, because if you read this paper, you know everything what I'm going to tell you. So there's nothing more than I have to say, which is than in this paper. And this is published, I think, 2 or 3 weeks ago. It's work of many people in our institute, together with people from Heidelberg, from Stefan Pieters' group, who helped us in the first 2 years, 2017, 2018, because we didn't have the facilities up to now, and the sequencing in those days was done in Heidelberg. After 2018, we did everything ourselves. So what is the promise of targeted cancer therapy in children? Well, you have all these patients that look similar, but are not similar. And if you do molecular genetic profiling, you see that all these patients differ. And the question is, can we give all these different patients a different treatment that is individualized? Give them their, let's say, the ideal situation of personalized medicine. Of course, this is an ideal picture, but we're slightly going there. And when we started our ITER program, we wanted to have, we had several goals. We wanted to implement the precision medicine. We want to evaluate what would be the clinical benefit of this program, and does this indeed look to work, do we work to personalized models? So the workflow is quite simple actually. So you start with the patient, you take a sample, a biopsy, and because our surgeons are very much involved in this program, we usually get enough material to do this. You do the sequencing and the bioinformatic analysis, and from there go to the data visualization. We do RNA sequencing, methylome profiling, and also germline whole exome sequencing. As a research, we do whole genome sequencing, but for this program, we used the data I just mentioned. And then the most important step comes, how to use these data. So we discuss this in multidisciplinary tumor boards, or molecular tumor boards. Every week, we have 3 molecular tumor boards. One for children with brain tumors, one for children with solid tumors, and one for children with hematological malignancies. And in this tumor board, we have the clinicians there of this specific tumor type. We have the diagnostic people. We have people from the trial and data center. We have and people from the research. So also the researchers who, let's say, work, for instance, on the brain tumors are there at this molecular tumor board. So they learn how the clinicians use the data, and the clinicians learn from the researchers. Most important are the molecular diagnostic experts from our diagnostic laboratory, and everything, all the diagnostics are in one hand, in one laboratory. And from this tumor board, we come to a treatment recommendation to a targeted therapy or conventional therapy. Do we do this on all patients? Yes, we do this on all patients, also upfront patients. But of course, in practice, all newly diagnosed children are treated according to standard protocols. So in between 2017 and 2021, we had 302 samples from 253 patients that had a primary high-risk tumor, a refractory tumor, or were at relapse. So we saw in this time frame roughly 600 newly diagnosed patients each year. So in together, let's say more than 2,000 patients. But from these 2,000 patients, only 253 belong to this category. And if you look to the stage of disease in which we did all this molecular profiling, according to age, the numbers differs a little, but most patients had relapsed cancers, first or second relapse cancers. And if you look from which type of cancer these patients come from, it's in different colors. I'm colorblind, so I can't see which colors are there, but I think this is blue. These are the solid tumors, CNS tumors, and hematological malignancies to start with. And then if you do the profiling, with the techniques that I just described, at the end you end up with possible targets of therapy that we categorize according to a certain system. Very high, which means very high likely to be a good target because it's let's say, abnormal in the cancer cells, and it has been proven that this abnormality plays a role in the tumorigenesis of this tumor. High means abnormal in the cancer cells and have been proven in another type of cancer to be important for the tumorigenesis, and then it goes down. In practice, only the very high, high and moderate are more or less suitable for targeted treatment. So if you then look by tumor type, neuroblastomas and high-grade gliomas are the most likely where you find a very suitable target for precision medicine. And for some other tumors, like osteosarcoma, the likelihood of getting a good target for treatment is relatively low. What are the results? Well, altogether, we found somatic, so alterations in the tumor in 90% of all patients, and a targetable abnormality in 82% of all these patients. With the clinical priority as stated here, so 6% only had a very high likelihood of being usable as a therapeutic target, 20% high and 26% moderate. So altogether, roughly half of patients from these 82%, so 40% overall, have a suitable target for precision medicine. A side effect of all these molecular genetic profiling was that we saw that not always the diagnosis was correct, and based upon this research, we changed the diagnosis and the treatment for 3.5% of patients anyway. Also, what we found was abnormalities in the germline, in the normal cells, in 60% of these patients, of which 10% had a tumor predisposition causative factor. So if you look to these germline variants, so in 10% of all patients we found an abnormality that was associated with the development or a predisposition to develop another tumor in future, which is very important information for the parents and also for the doctors. In how many patients did we really apply precision medicine? That, of course, is the most important at the end. Well, in 11% of all patients, we really applied a personalized medicine for these patients. And from those patients in which we used a targeted therapy, 16 or 18% had a very high target, so a highly targetable target, 46% high and 36% moderate. And for those patients in which no therapy was applied, still 9% had a very suitable target. And the question is, why did we not use these data for these patients? Well, the reasons for not using the data from the precision medicine program are listed here. And the most important four are on the lower side of this slide. First, because the disease stabilized by conventional therapy. And both the clinicians and the parents didn't want to change and didn't want to take the risk of getting a precision drug instead of standard treatment. Second was that parents refused, or the patient, him or herself, refused, because they said, well, it's enough. I don't want any treatment anymore. It's fine. Third reason was a progression of the disease. And malignancy evolved so quickly that we couldn't apply the precision medicine. And the fourth reason, that a drug was not available. And people from the Trial and Data Center are always there at the Molecular Tumor Board because for most drugs are not registered for children, but we also have all our contact with the pharmaceutical companies, and the people from the Trial and Data Center then get into contact with the pharmaceutical companies to get the drug on a compassionate use basis, and most often this is approved by the companies. This is a very important slide. Does it help precision medicine? Does it give you a benefit? This is not only data from our ITER program, but also from Stefan Pfiester's group, the INFORM group, that included in those days the ITER program. And I think it's the red curve, but if it's green, please, then it's green. I don't know. It's the upper curve, it's the survival curve for those patients that are treated with precision medicine. Drugs chosen based upon the Molecular Tumor Board meeting, while the lower curves are for those patients that are not treated with precision medicine and having the same genetic abnormalities, and the black curve is for all other patients. So there is a significant difference in survival between those patients treated by precision medicine. So in conclusion, comprehensive molecular profiling across all ages and all tumor types is absolutely feasible. Nowadays, we've included this in our standard care program, so it's done for all patients already at first diagnosis. And we combine it with drug sensitivity profiling on our organoids and PDX models for hemato-oncology patients. And we combine it, and this is not on the slide, with immunotherapeutic targets, because immunotherapy is coming up very quickly, so we combine it also with whether the cancer has specific antigens that we can target by monoclonal antibodies or by CAR T cells. And by having said this, I want to thank you for your attention and showing a picture of our center at upcoming Sun. Thank you.
Thank you. If somebody have questions, please, because it's no question in the chat. I would like to ask one question. If you have primary patient with high risk, you know that you will be lucky with results, but you know big percent you are unlucky. You will start with this thinking about this precise medicine for those ones?
No, we always start with the standard protocols that we have for newly diagnosed patients, but because the sequencing is now standard included in our primary diagnosis, we have all these data. So it is easier than to use these data if a patient will appear to be refractory to the first-line therapy, and then we don't have to wait several weeks for these results. So it takes about 3 weeks to have all these data from the sequencing and all the bioinformatics that you have to do before you get a result that you can discuss in the tumor board.
Okay, thank you. One more question, please.
I can hear you, but your microphone is not working.
So this frequency that you mentioned, 11% of patients do in the end receive target treatment. Is that similar for your groups, solid tumors, brain tumors, hematological malignancies?
Yeah, so the question, I will repeat the question for those who couldn't hear. So is the 11% of patients that we use the precision medicine, is it different between solid tumors, brain tumors and hemato-oncology? Well, it differs, and it even differs within, for instance, the brain tumors. There are several brain tumors which have a high likelihood, like the high-grade gliomas, and several brain tumors that you never find a therapeutic target. So it differs by tumor type, indeed, yeah.
And also by relapse frequency, obviously, because you only treat in the relapse situation, right?
Yes, we only treat in the relapse situation, yeah, yeah.
And one more question, please.
Yeah, I will repeat the question, yeah.
Okay, thank you, Rob. Very encouraging talk. My question will be a bit provocative, but that's a question which I hear in many of our conversations. So would you advise to continue the effort to advance molecular profiling if there is no next step available, so no targeted therapies are available?
Yeah, so yeah, we simply do it on all patients, and on beforehand you don't know whether you will find a target in these patients. But we have other work, and which has also been published recently, that show that the sequencing can replace a lot of our conventional diagnostics. So even if you don't use it for precision medicine, we use it for classification of patients for the diagnosis by replacing other techniques, which at the end makes the diagnostics not more expensive, but you get more data for the same price if you use the sequencing data. Not the whole genome sequencing, that's still quite expensive, but the RNA sequencing picks up, for instance, translocations much better than our conventional diagnostic methods.
Thanks, I'm very satisfied with the answer.
Thank you.
Okay, thank you. And we will continue with children cancer, and the next speaker will be David Gisselsson Nord from Sweden, from Lund University Cancer Centre, Division of Clinical Genetics. Please, floor is yours.
Thank you very much. I'm greatly honored to be invited here, and I think it's a perfect continuation of Rob's talk, because this will be largely about the diagnostic utility of whole genome sequencing and for children with cancer. So we are taking a somewhat different approach in Sweden. Sweden is a vast country if you compare this to the Netherlands, so a centralization to one spot is really not possible. So instead we have a national consortium called Genomic Medicine Sweden, and I represent the Pediatric cancer track in the Genomic Medicine Sweden. And we decided a couple of years ago, starting with the implementation in 2021, to actually establish nationwide access. So nationwide for us is 6 different centers spread across the country to whole genome sequencing and RNA-seq to pediatric cancer patients. And actually our primary intention of this was, because here the evidence was quite clear, to increase diagnostic precision and also refine the risk stratification of these patients. And then, yes, we knew that we would find drug targets, but we was also quite aware that this would be a minority of patients with the drugs available today. And we also was quite aware that most kids do super well on first-line therapy. But maybe there would be a few patients of interest where we could find drug targets. We were not too optimistic about that. We also have a streak where we identify hereditary condition by germline whole genome sequencing. We expected slightly above 10% to have such a condition. We knew that from previous studies. But there was also, this was not only implementation into healthcare. We also had a research track here where we decided to actually create a national resource of DNA and RNA sequencing for research use in a national repository where a researcher could apply to get both biological material and sequences from a child's tumor. And of course, this is we think, probably one of the absolutely first nationwide implementations of whole genome sequencing for an entire patient cohort. So we did not select patients really based on their stage or risk. Everyone was included if there was sufficient material, and sufficient material there was in this solid tumor cohort in around 2/3 of patients. We are still struggling with the last third of these patients. So the challenges in this case is that, again, we're a vast country, and not only that, it's also that we have a strictly regionalized healthcare system. So as a project leader, I cannot order anyone outside my own healthcare region to do anything. You just have to really lead by example and with moral leadership. You have no authority at all. You just have to say that if you do this, you will get money. And it will be good for the patients. And hopefully they will do it. The good thing is that pediatric oncology community in Sweden is very friendly. Before this project, there was, although quite a lot of competition between the different centers. We have the Karolinska Institute in Stockholm, we have the Sahlgrenska in Gothenburg, and you can imagine that there is quite a lot of competition going on. But this has kind of phased out quite extensively. So there is no top-down government steering. This is all done in a classical Swedish model of like decentralized initiatives. So you have to find the right person in the right center who wants to do this for you, typically in his or her spare time, you know. Logistic was a bit complicated. We had 3 sequencing nodes at the biggest university hospital, Lund, where I come from, in the south, Göteborg in the west, Stockholm in the east. And then we have to have some sample being sent. Methods were expensive, but that was the least of our problems, because funding was good in this case. Both private funding for the Pediatric Cancer Fund, and we also got additional government grants, because it was turned out to be a bit awkward for the Swedish government to have such a project, nationwide project, being sponsored only by a private foundation. Methodological expertise was required, but we could import a lot of that from academia. And then we had to build teams again. So in some, we have to get oncologists on board. So in some places, the problem was really getting oncologists on board, and in other centers, the problem getting pathologists on board. So we had to create all these regional teams with all the different specialties, including, of course, research nurses, and we also had in-house ethicists, and we had a lot of legal challenges that were taken care of. So we started out with actually taking care of the legal challenges and ethical challenges, getting all the permits to do this, and also to share data between healthcare regions. And then we set this system up, very similar to the Princess Máxima system. You have a dedicated pediatric surgeon who gets you a fairly good biopsy. It's checked by pathologists that you have at least 40% tumor cell content. You do nucleic acid extraction, make sure that you have informed consent, at least at the beginning. Now we actually start with just running this as healthcare procedure, and then if we get informed consent afterwards, it's good, and then it can be used for research. But we now include, actually, this is treated as standard of care, because we have found that the diagnostic utility is so high. Then patients are included if it's sufficient material, and then we do whole genome sequencing, blood, tumor, RNA-seq, and if it's a CNS tumor, we also do DNA methylation in one national center. We look at since we do whole genome sequencing, I just want to point out that we also look at the chromosomal copy number profile, which is very diagnostically informative in childhood cancer. It's super useful. We have regional molecular tumor boards, 3 of them, actually, and also a national board to which we can refer cases. And then this is reported out, actually, as part of the final pathology report. We integrate the molecular whole genome sequencing results, and then data is shipped off to a national data cloud. So what did we get from this? So this is a summary we did in April, when we had been running for 13 months. We had 118 patients including. We are now around at 160, because now we have also included all the leukemias, so it's also part of the project in leukemias. One thing that was really, really nice was that we had equal inclusion rate among the 6 national centers, which is so, and that was really, really good. It's kind of difficult to get. So they all had included patients in proportion to their patient burden, and that's really, really important in a national project like this. We caught diagnosis as in proportion to incidence. One exception was lymphomas, because as you know, for example, in Hodgkin lymphoma, the tumor cell content is maybe 1 to 2%, even in a diagnostic sample. You can't do that with whole genome sequencing. It's not possible. We found genetic variants that had clinical associations, meaning that they had at least a diagnostic informative value in 91% of the included cases, and drug targets. And now we talk about kind of high confidence or very high confidence in 24%. So it's surprisingly similar to the Princess Máxima experience, actually, where we went up to 26%. But this is an unselected cohort. So it's pretty much the same. And we find it like expected targets, ALK and track fusions. So that looks good. The thing with whole genome sequencing is kind of you find interesting and unexpected things. And so I'm going to show you one of those cases here. So this was an embryonal rhabdomyosarcoma. It's a skeletal muscle-like tumor that was in this case sitting in the prostate of a small boy. And in this case, by whole genome sequencing, we found something which was very, very rare in these tumors. We happened to find amplification of a gene called HER2 or B2, which most of you probably recognize because it's a famous gene from breast cancer where it's amplified. So this is like the surprises you get with a whole genome sequencing approach, that you find these strange cases where you actually, this simple amplification that can be proven also by FISH and immunohistochemistry, actually, if this patient relapses, which is pretty high risk because surgery was not radical, we know that you can use Herceptin from breast cancer to try to treat this tumor, which is really useful. Another example actually where sequencing came out to be really interesting and actually revised the diagnosis. This was a spindle cell tumor in the neck of a small girl that we thought first was an infantile fibrosarcoma. It really had high NTRK expression by proteins, but when we do sequencing, we realized that there was a really, really complex rearrangement going on between chromosome X and 18, creating an SSX, SS18 fusion. So this was actually a synovial sarcoma, and treatment changed completely. Another case, neuroblastoma, diagnosis in this case was super easy, but we had a really, really complicated things going on in chromosome 2. Those are the green spots that you see up there. And by the whole genome sequencing, you can resolve that, saying that this is actually a complicated amplification, including a fusion gene between ALK and DUSP3, and this patient is then eligible for ALK inhibitor treatment. And so on, another complicated case. But the challenges in the end, what challenges did we find when we did this on a national level? First of all, cash is a big challenge here. These are expensive methods, so each patient here costs around 5,000 euros to run. Right now we depend on specific external sponsorship. We have a political challenge ahead to trying to see to what extent will this be taken over by the budgets of the healthcare region? It's like a political battle we have in front of us, because we know as the diagnosticians that this is super useful, but is it really cost efficient? That's the question. As you saw from the very interesting INFORM study data, yes, there is prolonged survival, but do we know that it actually saves lives? Do we know that it's cost efficient from a national perspective? These are open questions. Another question is, of course, time, again. We have to have more time to study. Will we get time? Can we buy time for this to know if it really quality of life and overall survival increases? We don't, it's completely unknown. And also evolution, of course. Because one of the main reasons that you saw that even with the fantastic INFORM study, you get around 100 plus extra days of survival, but all the kids died in the end, right? And one reason for that is because you have tumor evolution. Just showing you one of our patients as an example. This last minute, small boy, he had his second relapse of neuroblastoma. We had analysis done at 2 different sites. One was a giant tumor he had in his mandible. The other place was a tumor he had in his bone marrow. And in the top one, you can see that there is this amplification here of the ALK gene, well-known drug target, also mutated, but he also had a lingering bone marrow metastasis here without the drug target. So he got lorlatinib, he got treated for his ALK mutation and amplification. He had remission for 6 months. But you can imagine what's lying there behind, right? That they had this non-responsive clone lingering in the background. And sure enough, after 6 months, he had explosive relapse and died from his disease in this case. So this is the family tree. We do a lot of these evolutionary studies in the setting of this project when we do multiple sampling. You can see that in this case, so this is what we treated. This is the ALK amplified part of his tumor's family tree, but what killed him is actually up here without ALK amplification and mutation. So a big challenge, I think, for all of us in precision oncology and precision medicine is kind of how can we lure cancer, how can we trick cancer evolution into different dead ends. But that's another discussion for the coffee break in the end, I would say. So to summarize this, clinically informative variants, especially of diagnostic use, actually, so we could actually, in the majority of cases, could be better sub-classified with genomic data, really. So we have really informative data by these expensive methods, whole genome sequencing and RNA-seq. Even in this unselected cohort, where actually we included all kids with sufficient material. Sustainability is still a challenge. Is this financially viable from a health economic ethical perspective? It's an open question. And also we have to kind of, in the long run, to make precision medicine work, somehow compete and handle tumor evolution. The biological challenge is really still out there. So we have to also innovate, not only new drugs, but also new treatment concepts to have precision medicine going. But that's a long discussion. I will save it for a coffee break. If anyone wants to discuss evolutionary strategies, I'm happy to talk to you. Thank you.
Thank you. Colleagues, do you have a question to Professor?
Can I ask a question? You talked about the finances. Suppose you would only do RNA sequencing. What would, and forget about whole genome sequencing, what would be the cost then?
They would be just 1/10 of the cost.
Yeah.
But we will miss a lot of diagnostic information. On the other hand, we expect the price of whole genome sequencing to go down. So we think that if we just keep doing this, then it will be so cheap that it's easier to argue to the politicians that we should do this. Because we see every year that already from the start of the project, the price went down from 7,000 euros, which include like all the bioinformatics and data storage and everything, to 5,000 euros. So it's still going down. Yeah.
Okay, thank you.
One more question, please.
With what coverage you are running the whole genome sequencing, so because you have to balance out always the identification of mutations versus the price, which is
yeah, minimum of 90 times coverage. Usually our average coverage is around 113.
That's already a lot.
Yeah, it's expensive, but still. Thank you.
Okay, thank you. And the next speaker will be Alwin Krämer, full professor at University of Heidelberg, director of Clinical Cooperation Unit Molecular Hematology Oncology of the German Cancer Research Centre, Department of Internal Medicine, University of Heidelberg. The topic is very interesting for all of us, cancer of unknown primary. Please, Professor.
So let's see whether I get this working. So thanks for, first of all, the invitation and also for the kind introduction. This talk will differ from the earlier two ones in two aspects. First, it focuses on a specific disease that you might not even have heard of, cancer of unknown primary. It's comparatively rare. And second, this is an adulthood cancer, no pediatric cancer. Okay, so in brief, what is this? So it's a situation where a patient comes to see a doctor with metastases and the responsible primary tumor responsible for the metastatic seeding cannot be identified in those patients. It's a rare condition, 3 to 5% of all malignancies, and for the talk you don't need to know a lot about it, but one important point here is old autopsy studies have shown that in those cases where the primary could be identified, which is a minority, and in those patients the primary is rather small, it is spread all over the organs of the body. So it's not a specific cancer type, but it stems from different organ systems. Very frequently the lung and the pancreas, but also other organs are important. So why is
reflecting on cancer of unknown primary relevant to this audience, to people who think about implementing precision oncology? I think this is one of the reasons for that, because this is kind of a paradigm of tissue agnostic cancer. So you don't know the primary, so therefore it has to be tissue agnostic, and tissue agnostic treatment by targeted options, treatment options, is one of the goals of precision oncology, obviously. The second reason is about 85% of those patients fall into a prognostically very unfavorable group, meaning that even within clinical trials, for patients that are treated with conventional chemotherapy, the survival is really poor. So in clinical trials, as depicted here, it's 9 months. Outside of clinical trials, so in the real world scenario, it's even poorer. It's around 3 to 5 months. So there is an urgent need to improve on that in this cancer type. Precision medicine, or the startings of precision medicine in cancer of unknown primary, given this extremely poor prognosis, date back for quite a while. About 20 to 25 years ago, people started to use a type of precision medicine, in this case gene expression profiling, to learn on the origin of cancer. So a piece of tissue was taken, gene expression profiling was done on this, and then the result of that gene expression profiling was compared with large databases of gene expression profiling results from cancers with a known primary, in order to then extrapolate from there, this cancer of unknown primary is likely a, I don't know, pancreatic cancer or breast cancer. And then treatment of chemotherapeutic treatment on this specific type of cancer that was found by gene expression profiling was tried. However, 2 recent randomized studies have shown that this is not the way to go. When you look here, so these are the results of 2 gene expression profiling-based primary identification trials that have been randomized between organ-specific or proposed organ-specific treatment versus unspecific CAP chemotherapy. You can see that here. So, sorry. These are the 2 unspecific treatment schedules that were applied in the control arm, and these are the schedules that have been applied in the organ-specific arms. And unfortunately, here on the right side of the slide, you can see the results. The curves are, in both trials, completely overlapping. So there is no progress by trying to identify the primary. The same is basically found, and this is an important slide as well, in a large meta-analysis on that. Here you can see the results of the 2 trials that I've just shown, so no benefit for organ-specific chemotherapy. And these are the results of earlier trials, non-randomized, that suggest the benefit of organ-specific treatment, but that could not be refound or recapitulated in randomized trials. So this is a further argument of careful examination on what you are doing, not just non-randomized trials and then suspect there might be an advantage. This is obviously not good enough here. So now we are approaching what we are actually talking about here, namely precision oncology. People have started sequencing CAP cancers, so cancers of unknown primary, similar to virtually all other types of cancer, and the results were not so different, not unexpected, not so different. So this is the mutations that you can find in cancers of unknown primary. This is the frequency of their presence, and you can see the usual suspects here in front: TP53, CDKN2A, KRAS. These are the very frequent mutations that can be found in many patients, and then there is this long tail plot with many, many additional mutations that can be found, and the frequency is decreasing from left to right. And in blue, you can see those mutations, not important which ones these are, that can, at least theoretically nowadays, be targeted by drugs, because drugs become increasingly available for different types of mutations here. And this is the same in cancer of unknown primary as it is the case for other cancer types. In Heidelberg, we have performed a large trial, it's called the master trial, where we, outside of a randomization and retrospectively, do sequencing of patient material from patients with solid tumors that have relapsed after first-line treatment. And after that, those patients then, if a target is available, receive targeted treatment. And here in brief are the results of that trial that have been recently published. And when you look up here, you see unknown primary. So this is cancer of unknown primary on both sides. And you see that immediately after non-small cell lung cancer, CUP patients outside of a randomized trial seem to benefit quite a bit from targeted treatment options. This is also because the results of conventional treatment are so poor, but both with regard to best response after molecularly guided treatment, and when you compare progression-free survival rates after molecularly guided treatment versus progression-free survival after initial chemotherapy, then patients with CUP do quite well. And this led us to the suspicion that this is an entity where it's worth trying to compare really targeted treatments with conventional chemotherapy in a randomized setting, and I'll come back to that in a few minutes. Also, immunotherapy, it has been mentioned in the last talk already, seems to benefit patients with cancer of unknown primary. These are the results of 3 rather small trials, second-line treatment of patients that failed conventional chemotherapy with immune checkpoint inhibitors, and all 3 trials seem to show the same, namely a response rate of around 20% of those patients, which is for cancer of unknown primary quite a bit. And also predictive markers of response to immune checkpoint inhibitors. One is expression of something that is called PD-L1, and the other one is called tumor mutational burden, TMB, seem to be relevant in this cancer entity. Here you can see on the left-hand side that PD-L1 expression predicts the response rate to immune checkpoint inhibitors, and on the other side, on the right-hand side, you can see that tumor mutational burden predicts survival after treatment with immune checkpoint inhibitors. And this is actually one of the big problems in precision medicine. You don't know, except for the presence of a target, which patients will respond, and therefore predictive markers are urgently needed. And in the immuno-oncology field, those 2 predictive markers, PD-L1 and tumor mutational burden, seem to work at least to a certain extent. Another way to predict response is not before starting therapy, but during therapy. And there is something called ctDNA, so for circulating tumor DNA, and you can measure within the blood of a patient whether ctDNA, tumor DNA, is present or not. And the level of this ctDNA predicts response to, in this case, immune checkpoint inhibitor treatment as well. On the left-hand upper side, you can see that those patients that do respond to immune checkpoint inhibitors have lower levels of ctDNA, and on the right-hand side you can see that those patients depicted in yellow they have a decline of their ctDNA levels already after the first cycle of treatment, and those are the patients that do respond to the treatment, whereas the others, where no decline of ctDNA levels can be found, do not respond to the treatment. And in the lower part of the slide, a single patient is shown that very impressively shows this fact. When you look here, so in the upper part, you see the diameter of the metastasis in computed tomography images, and this is the level of ctDNA. And when you then conventionally check whether the patient has responded to the treatment by imaging, there seems to be, at least initially, no response, but the ctDNA level immediately dropped to almost 0. And finally, the metastasis then also shrunk. So this is another option of predicting response to treatment by measuring ctDNA. So now to the COPISCO trial. I mentioned that briefly already. This is a trial that in patients with cancer of unknown primary, in a randomized fashion, compares targeted treatment on the one hand versus conventional chemotherapy on the other hand. When you look on the left-hand side, patients with newly diagnosed unfavorable cancer of unknown primary are included into the trial. All of those patients receive sequencing, so panel sequencing, and then they receive 3 cycles of induction chemotherapy. After that, those patients that respond to that treatment, at least with a stable disease, are randomized into an experimental arm and into a standard arm with continued chemotherapy down here. The experimental arm, and this is also important in that trial, consists out of 12 strata with different targeted treatment options depending on the result of the initial panel sequencing. So a patient that has an IDH1 mutation, for example, receives an IDH1 inhibitor. A patient that has an FGFR1 or FGFR2 fusion receives an FGFR inhibitor, and so on and so forth. And in the end, we will check whether patients receiving one of those targeted treatments do better as compared to chemotherapy. And as I mentioned, we learned that the hard way for the gene expression profiling data that I have shown. We needed a randomized trial to really be able to judge that this doesn't lead us anywhere, whereas the non-randomized trials that were performed before that didn't give that answer. Okay, as I said, CARP is a relatively rare disease, so therefore this trial, the COPISCO trial, is a really huge endeavor. It's an international trial. 34 countries are involved in it and more than 150 local sites. And in order to recruit around 700 patients, we screened more than 1,400 patients already. We are almost there. Most of the patients of those 700 that were initially intended have been included, and we are looking forward to the results and hope that precision medicine in this entity will help to improve the poor prognosis. This is one case report. Briefly, patient with a BRAF mutation did receive, after the initial 3 cycles of induction chemotherapy treatment with vemurafenib and cobimetinib, so a BRAF and a MEK inhibitor, and she entered complete remission and stayed there for more than 2 years already now, which is really rare for patients with CAP. And 2, I think, final slides. In addition to improving single patient care, we also learn to molecularly classify CAP syndrome and learn where the cancer really stems from. We have here several clusters, 10 in total. Cluster 2, mutations shown on the right-hand upper side, mutations in FGFR2 and BAP1 found, highly suspicious of cholangiocarcinoma, and in the lower panel, NF2, SETD2 mutations, highly suspicious of renal cell cancer as the origin of the CAP syndrome. Can maybe skip that. And the final slide, It was mentioned earlier that germline mutations predispose to cancer development. We have looked into patients with CAP syndrome and first found that about a quarter of those patients had an earlier cancer during their life already, and the second cancer was by the pathologist betted that it was not a relapse of the initial cancer. We then did comparative sequencing of the initial cancer versus the proposed CAP syndrome and found two scenarios. One scenario up here, identical mutation patterns between the initial primary cancer and then the proposed CAP syndrome, arguing strongly for a relapse as the reason for this CAP syndrome. And in the other third of patients, we found indeed different spectra of mutations. So these are two different cancers, obviously, but presence of germline mutations, thereby predisposing to the development of different cancers in single patients. So in summary, CAP may serve, as I said, as a tissue-agnostic paradigm for precision oncology approaches in primary metastatic cancers. Platinum-based chemotherapy in those patients is simply not good enough, so we have to do better, and precision oncology is one option to do so. Primary identification by gene expression profiling as a first historic attempt of precision oncology failed in this type of cancer. Second-line immune checkpoint inhibitor treatment seems to be beneficial, especially in those patients where we have a predictive marker, in this case, tumor mutational burden or PD-L1 expression. The large CAPISCO trial, international trial, tries to show or to prove that precision oncology is better in a randomized fashion as compared to standard chemotherapy. Antecedent malignancies, as I've shown you in patients with CAP, suggest either a hidden relapse or germline cancer predisposing mutations. And finally, the mutation analysis of CAP specimens allows for molecular classification and identification of pathways responsible for early metastatic seeding. This is the German Cancer Research Center, where the research part of what I've shown is done, and in the associated hospital to that, so the University Hospital of Heidelberg, many of the patients that are included in this talk have been treated. Thanks for your attention.
Questions from the audience or from people at home or wherever they are, actually? Yes. Could you please identify yourself?
Good presentation. I'm Jukka Kanerva, pediatric oncologist in Helsinki. In the CAPISCO trial, you randomize patients who have had response to cisplatinum. Is it easy to convince the patients to leave cisplatinum and go to targeted therapy?
Is it easy to convince patients to?
If they are randomized from the treatment which they have had response to?
Yes, actually it is easy because most patients do know that responses, so even if you respond to a platinum-based chemotherapy, then your prognosis is still extremely poor. And most patients are really looking forward to receive targeted treatment in contrast to continue with a platinum-based chemotherapy.
Thanks.
Can I ask a question? In the CAPISCO trial, so you randomized between standard treatment and precision medicine based upon different profiles. And these profiles were made before you started doing the sequencing. And by sequencing these tumors of unknown origin, did you find different pathways that you didn't anticipate on?
You mean new pathways that were unknown to us beforehand?
Yes.
Not really, because before the trial started, a handful of larger sequencing studies in CAP have been published, and we knew beforehand what is to be expected when you sequence CAP patients.
Okay.
And this is also the baseline, why we set up the study in the way it is set up and why we use the drugs that we use in the trial, because we knew beforehand which mutations are more frequent and which should be targeted accordingly.
Okay. Okay, thanks. Yeah, we can't see, I'm looking at the technical people, we can't see whether there are questions from, no, there are no questions. Thanks. Okay, thank you. We continue, yeah, you may give an applause if you like. Always nice to have applause. We go ahead with Martin Zoche, who is a director at the Molecular Tumor Profiling Department of Pathology in Zurich, and he will talk on homologous recombination deficiency testing.
Thank you very much for this very nice invitation and the hospitality here in Latvia. And I have to say I was very impressed about the talk of the young patient earlier today in the morning. And I think this remembers us all the time why we are here and why we are doing this molecular tumor profiling. In the end, it's a patient which counts. But I want to switch now a little bit gears and go away from the mutations in the tumor, which we can identify for treatment, and going more into the direction of biomarkers, because we also can identify biomarkers during such a panel testing. And such a biomarker is really the important thing also, which can lead to targeted treatments. One of these biomarkers is the HRDness. This stands for homologous recombination deficiency, and this can be caused, for example, by the BRCA genes, but also by many other genes. And I want to give you an introduction into this biomarker, as well as showing you a little bit how you can diagnose these biomarkers, how you can do the assays to identify if a patient has such a HRDness. The BRCA genes are well known, so this is a very old publication, more than 30 years old, showing that BRCA genes are responsible for specific cancers. And we know today that the homologous recombination deficiency is caused by these BRCA genes. We know also that BRCA genes, that the mutation in BRCA genes can be autosomal dominant, be hereditated, and that the risk if one of the parents have a BRCA gene for children also is very high, and that these these patients can become patients as well. So we have different incidents for the different mutations in BRCA genes, and for example, it's very high incident rates in women to get breast cancer or ovarian cancer, but men are also affected from this because pancreatic cancer and prostate cancer is quite a common cause of the BRCA genes. We observe in even more cases that it's not the BRCA mutation which is causing cancer, but it's the HRDness. And I will show you in a minute what HRDness really means and how we detect HRDness. So in 1 of 4 women, a BRCA mutation is identified in advanced ovarian cancer, but in 1 of 2 women, we already see the HRDness. And this can be caused by other factors as promoter methylation, other HRR genes which are mutated, and still today unknown causes. Here you see the cascade, the pathway, how the recombination deficiency or how the recombination happens in our cells during cell division. And you can imagine that not only the BRCA genes, which are here in the central position, but other genes, when they are affected and mutated, also can cause a deficiency so that these cells cannot divide anymore correctly. And these deficiency of not dividing correctly in the end causes cancer. We know 3 biomarkers or 3 observations we can make of the HRDness. And one is the loss of heterozygosity. the telomeric imbalance. Here I will only show that in the end the telomeres are affected and can be not divided anymore correctly. And the third one is a large-scale transitions. And the LOH and the LST I will show you on the next slide. So what is loss of heterozygosity? It simply means that we have small pieces of DNA in the chromosome, less than 15 megabases, which get lost, which are not repeated and correctly anymore. And we simply count these areas of the genome, and then we can identify by adding this together and coming up with a percentage of genomes which are not correctly divided anymore, and we see here a loss. And this loss of heterozygosity, if this is a certain value, we say around 15 to 16% in ovarian cancer, then these patients also can be substituted with a PARP therapy. Large-scale transition, in contrast, are the pieces which are larger than 15 megabases of genome. And here we are not only counting the losses, but also the amplifications. And adding up these two things together, we are coming with a biomarker for large-scale transitions as well. And this is how it looks in real life. So here we have a patient with quite a stable genome. So this is a tumor patient where almost we do not see not very much indeficiency. And in contrast, therefore, we have an unstable chromosome patient. And here we see a lot of losses and amplifications of the chromosomes. And that means that such a patient here might receive a targeted therapy, a PARP inhibitor therapy. We can detect these loss of heterozygosity and the HRDness by different means. So one is that we are going into OncoScan and we can make arrays to detect the SNPs. We can do targeted therapy, and this is the preferred method, as well as shallow whole exome or whole genome sequencing. Why we are using targeted therapy? Because in addition to these biomarkers, we can of course also detect the mutations which also might be or can be treated, or where we have treatment options. So therefore this is for us in routine diagnostic the preferred method, because here we are combining the targeted mutations from EGFR genes, from other genes, together with the biomarker of HRDness. We have these 4 different alterations, and we are looking in a targeted sequencing in the end just to these 4 classes of genomic alterations. And we prefer at the University Hospital in my laboratory the hybrid capture sequencing method, because hybrid capture sequencing has an advantage against amplicon sequencing, which is another method, because here we are sequencing into unknown areas of the chromosomes, and this has advantages for us in identifying how the chromosome or how mutations are set. And I will give you 2 examples for this. We can skip this. One example is shown here. So what we are seeing here is that we have a mutation which was not identified before in the exon 23 of the BRCA chromosome 2. So in the initial analysis, in the initial diagnostic, this patient was seen as a healthy patient regarding the BRCA chromosome. When we have analyzed, we identified another adenine here in the sequence, which causes in the end a termination of the BRCA gene. And these homopolymeric regions are much, much better to be identified in the hybrid capture method than compared to the amplicon method. So the message here is really that you have observed very, very carefully what kind of method you want to use once you're going into the diagnostic of genes. Another example, exons can be flipped around. So here is the BRCA wild type shown, and here is the exon 15 flipped around. And this flippage, of course, destroys the amino acid structure of the protein, but this can be only identified if you sequence into the intron regions of both sides as well, to identify such a mutation. It's not possible just to look into the exon sequence to see such an alteration. And of course, this has a huge impact because these mutations are, in the end, ARUP5 mutations. ARUP is a database at Salt Lake City where we look up all the different BRCA mutations, and we can see if such a mutation is a highly pathogenic mutation or a benign mutation. And this gives us an overview about the assays we are using mainly in Switzerland in the different pathologies. And on the right side, the three assays are the hybrid capture assays, which are really preferred assays if you want to identify HRD-ness. And for at least two, maybe in a few months, three assays, all three assays have also clinical validated data, which is an important aspect as well, because only with those you can really show the efficacy in a patient for the clinic. Just to give you two patient examples, I have here two patient cases. One is a woman, 49 years old, with ovarian high-grade serous carcinoma, and she knows the tumor since 2009 was the first diagnosis. Then after several years maintenance therapy, she received a relapse and progression in 2018. The breast metastasis was identified in 2020. It was already known that she is carrying a germline BRCA mutation, so she was treated with PARP inhibitors. But this doesn't work anymore very well. She received chemotherapy in addition, and we did a foundation test, a comprehensive panel test in 2020. And here you see the results of the test. So what we can show that here you have a high loss of heterozygosity. That means that this patient is eligible for a PARP therapy as well. And you see a BRAF mutation, which is quite rarely to be found in ovarian cancer. And the tumor board has decided that the BRAF mutation has to be targeted first before going on a PARP therapy. So here, this is a very nice example to show that in addition, with such a comprehensive panel testing, you see all the options, all the different mutations the tumor carries to then discuss best what is the right therapy option for this patient. Another example is a 90-year-old male, very fit actually, with bone metastasis of prostate cancer. And in the very beginning, Alwin, you will see, this couldn't be really identified as a prostate cancer because the PSA levels were very low, and the radiography of the prostate has really shown that there is a prostate cancer. So we have looked into the bone metastasis of the spine, and what we identified was a BRCA2 mutation, and this shows also the beauty of comprehensive panel testing. We have identified this as POP mutation, and this is not a targeted, treatable mutation, but we know when we see a SPOP mutation that the chances that this is a prostate cancer is very high, because this is a gene which is typically mutated in prostate cancer, but not in other cancers. So we really could confirm the diagnosis of prostate cancer for this patient. So the BRCA mutation, and this was a surprise, was also a germline mutation. We have transferred the patient to human geneticists to identify and could confirm that this is a germline mutation, and of course this has also implications for the relatives and siblings. So it was decided to start with a bicolantamide therapy, the classical androgen depletion therapy, but then later on the patient switched to olaparib, to a PARP inhibitor, as a next therapy option. Just to give you a take-home message, so several sequencing assays are available, and you really have to look into what is the best assay to detect, to answer your question you have to the test. You have to compare between amplicon-based testing and hybrid capture testing. And the hybrid capture technology detects definitely much better the HRDness of cancer patients. With that, I want to conclude. So this is my team, and we have also a lot of fun. But without all these people, all the work is impossible, because we have a lot of people working in the lab, as well as bioinformaticians who are then analyzing the data we are generating to come up in the end with the reports to the patients. Thank you.
Questions? Yes, please, Alwin.
So why is this seemingly arbitrarily distinction between LOH and large-scale transitions at plus versus minus 15 MB made?
Yeah, that's it's a little bit still, we still need data. So we know that LOH in ovarian cancer works quite well with, or PARP therapy works quite well. For LST alone, it's already questionable. So this is also always a vivid discussion we have in the tumor board when we receive this data and discuss if a patient who just have high LST value but no LOH or no BRCA mutation, if a PARP therapy is really worthwhile to move forward with. Here we are still lacking the data, and this is a big problem, yes.
And similarly, in many trials where a PARP inhibitor is applied, results seem to differ widely between different reasons of HRDness. Any idea why that might be the case? So the classical PARP mutations seem to respond well, whereas other mutations and other readouts for HRD seem not to work at all.
Absolutely. I mean, we even see that, for example, if you have a BRCA mutation, we do not see HRDness. So even that does not have a one-to-one relation. Sometimes we have a RUT52 mutation or something else, ATM mutation. Sometimes we see a high LOH, sometimes we do not see it. This is still work ongoing. Clinical trials are needed to better understand the pathways and why specific patients have a high LOH, and therefore they have a response to PARP inhibitors, and others do not, even though they carry the same kind of mutation. Yeah, completely agreed.
Okay, no further questions. Thanks for your presentation.
Thank you.
And the Finnish people will finish this session with two speakers. Don't know what that means, but Juha Kononen, the Chief Clinical Director of DKFZ Cancer Clinic, will speak on precision oncology in clinical practice. Not only the successes, but also the challenges, I see.
Thank you. Thanks for the invitation to give this presentation. This sounds like a pretty big topic for a 15-minute talk. So I'm going to try to give an overview from the oncologist's perspective on some of the key components and issues that I think are relevant at this time. So otherwise this would be a 2-hour talk. DKFZ Cancer Centre that I come from is the largest private cancer hospital in the Northern Europe. We mostly treat Finnish patients. We are based in Helsinki, but we do have patients coming from 40 different countries, including the Baltic countries as well. An important point to make is that we treat adult solid tumors, so no pediatric tumors. Here are my disclosures. To really think about what an oncologist needs to see, basically we need to know what is the diagnosis and relevant cancer classification, the accurate cancer classification. And in order to be able to do that, we do definitely need biomarkers. We need better biomarkers. We need, first of all, prognostic biomarkers to assess the risk for overall survival and think about different treatment strategies. Then we need those diagnostic biomarkers. Most of these biomarkers that are giving valuable information these days are genetic biomarkers, as we have heard in the previous talks. Then we really would like to have predictive biomarkers that would predict the response. And there are two kinds, positive predictive biomarkers as well as the negative predictive markers that suggest that you should not use a certain treatment strategy. Finally, predicting the toxicity would come from pharmacogenetic analysis, which you would get once you do whole genome sequencing. For an oncologist, for a clinician, it's also very, very important to know where the cancer is. And here we have these days these technologies that allow us to do that. There are these CT scans, whole body CT scans, MRI scans, targeted MRI scans, and various different techniques of MRI scans to really pinpoint the anatomy and localization of disease. And with MRI, you kind of step into the functional side as well with diffusion, magnet MRI imaging technologies. But finally, the PET scanners and PET tracers gives us the functional, metabolic, and molecular analytics that advance the precision medicine these days forward, and is the best kind of imaging technology in many situations. In order to make these treatment decisions, we have to consider also comorbidities and vulnerabilities for each and every patient. We would definitely want to see more effective treatments. That's while we have good and improvement treatments, we need better ones. And finally, for precision medicine approach, continuous monitoring and of response and keeping an eye on how the cancer evolves is hugely important. But we have had plenty of nice success proof of principle cases on how we can advance oncogene-driven cancer treatment forward. And perhaps one of the examples is in the non-small cell lung cancer, where this concept of using biomarker diagnostics to find the right drug for the right patient really has worked. So as a case in point, ALK fusion positive non-small cell lung cancer, the prognosis was really dismal before the discovery of the targeted therapies. The 5-year overall survival in stage 4 disease used to be 2%, very dismal. But already in the early stage of first ALK inhibitors, this improved markedly, so that the overall survival is over 50%. And actually the last year data showed that the median of our overall survival is close to 7 years, and we are continuing to see better and improved therapies to target ALK fusion positive lung cancers constantly. And this has really changed oncology in the adult cancer treatment setting as well. In many cancer types, we are seeing more patients being alive and more patients surviving with cancer, and cancer becoming partially a chronic disease. But this has not happened in all cancer types yet. We have had good progress in some, but there are still those very poor prognosis diseases like pancreatic tumors, where no real progress has happened over the last couple of years. So what are the challenges in precision oncology? One view to that is to take a look at what we know about the cancer driver genes. And one way to visualize this is kind of see the target genes in a hierarchical way, that we have a lot of cancer driver target genes that are mostly those tumor suppressor genes that are very commonly broken and the drivers in cancer, but those are not currently targetable. Then we have these alterations like the ALK fusion that is currently very, very targetable, but unfortunately rather rare. Then we have a mid-tier of alterations like FGFR and KRAS G12C mutations that are somewhat targetable, but we don't really know yet on what kind of combinations we need to use to target those diseases. So that's one aspect of this. The second one is that once you have a strong driver, it can be diagnostic for a rare disease subtype, like a breast secretory carcinoma with the NTRK fusion gene, or infantile fibrosarcoma for NTRK fusion gene. And then this can be very, very rare in a common cancer type. So what that means in the clinical practice, that we can't really restrict the sequencing and looking for these drivers based on the organ of origin. I would much like to see that we treating adult patients would have similar opportunities and similar routine practices as our pediatric, as our colleagues treating pediatric patients have. It was wonderful to hear that this is what's done in pediatric setting. It's clear that this should be done in adult setting as well, but we have so much more adult patients that it hasn't translated into routine clinical practice yet. Finally, looking at the drivers, the solid tumor genomes tend to be complex. On average, there's between 1 to 5 strong driver genes in every cancer. But there can be very complex combinations of those. So one cancer cell may have these 1 to 5 driver gene alterations, but a large cancer have millions of cancer cells that have many different combinations of those driver genes as well as the passenger genes that can change how the tumors really respond to targeted therapies. So currently, how many patients benefit from on-label targeted therapies? In 15 years ago, this was a dismal number, less than 1%. In 2018, this was already close to 5%. Now we are making true progress since on adult tumors, roughly 14% of patients can have targeted therapies. And a big change here is probably coming also, case in point KRAS, that is one of the oldest known cancer driver genes, but back in 2020, after almost 40 years of research and 40,000 of publications, there were still no drugs in clinical practice. But that changed with the G12C mutation that became targetable, and the drug has entered clinical practice already. And once you know how to do it once, this of course spurs a lot of research interest and renovates the efforts to target KRAS. And I believe that there are currently many different KRAS targeting therapeutic approaches ongoing, and those might enter the clinic in a few years. And this will then give us a new chance to really start treating diseases like pancreatic cancer that is really KRAS-driven cancer. One very promising avenue is also antibody drug conjugates that open up a lot of other cancer types for targeted therapy approaches. Case in point, recently triple-negative breast cancer that has been successfully treated with sacituzumab govitecan, targeting the TROP2 antigen present frequently on triple-negative breast cancers. In practice, what's the problem also when treating patients? Everybody knows that these single-agent targeted approaches tend to fail in 6 to 10 months, or maybe in a good case, 14 months. When you have combinations like BRAF and MEK inhibitors, you can have a bit longer responses, but eventually, almost always, the targeted approaches fail at some point in time. It's kind of, if you take the evolutionary perspective to this, makes sense, because biological systems have evolved to adapt, to maintain homeostasis. So there are almost always backup systems available. And once you block a pathway at one point in time, then it will become reactivated at another point in time. And if you have a cancer that has high mutation rate, it has an accelerated evolutionary pathway, it will happen faster. So that's an issue that we need to address somehow. We do know a lot about cancer these days, and this is one of the famous papers about the hallmarks of cancer that was just recently updated. So now we know that we have 14 key pathways or concepts, and this really explains the biology in very nice fashion. But the difficulty is that we don't have any metrics to these nice concepts yet. We don't have systems that would quantify how the cancer is gone wrong on all these 14 different key aspects. But this also makes it more understandable that why targeted single pathway approaches usually fail, because there are 13 other pathways that are wrong in cancer. So that leads us to the emergence of systems biology, and we still lack a lot of techniques in clinical practice that would allow us to do systems biology in that way. There have been some interesting approaches and examples like this recent science paper about the human cell atlas, where there was a transcriptomic profiling of the various different cell types in the normal breast tissue. Long story short, even in breast there are 12 different cell types that can exist in 47 distinct cell states. And in normal tissues, those cell state transitions enable a lot of functions in the normal biology, but in cancer, the cell state deregulation obviously contributes to the disease onset and progression and drug resistance. So, in order to move forward, in order to do better in precision medicine, this is the current state of the art, that we do gene panel sequencing, we do some immunohistochemistry assays and digital pathology, and then discuss these in a molecular tumor board setting. But in the future, we would like to see this going to the next level. We would like to see taking account into the tissue context and morphology and utilizing AI technologies to guide the tissue phenotyping, for example. And organoid models and drug response testing should also be utilized in those molecular tumor board discussions, as we heard earlier from the pediatric colleagues. Many of the things that I showed are kind of next level, next step, but what's currently in clinical practice in precision oncology is first of all techniques that allow more accurate diagnostics, as well as tumor monitoring. And PET-CT scanning is one of those techniques that allows us to pinpoint the metabolic activities in various different metastatic sites, and we have a continuous improving array of different PET tracers that allow us to glimpse into the glucose metabolism or bone function or estrogen receptor signaling or other features, functional features of the metastasis. Then the one currently very usable tool is the ctDNA that we also heard in the previous talks. All those metastatic lesions share ctDNA in blood, and then we can use that for many different purposes. Currently, what's done in the clinical practice is to use the ctDNA in the high disease burden setting, where we can utilize this for the dynamic monitoring of clonal evolution and find predictive markers when tissue testing is not feasible, and identify resistance mechanisms. We normally do something like 300 liquid biopsies a year at our hospital, for example. What we next want to do is to refine these assays into even higher sensitivity, so that we could move this technology into the low disease burden setting, where we could use it for early detection and cancer screening and detection of recurrence and finding the minimal residual disease, where this assay technology could guide us in choosing whether or not the patient needs a more intense treatment strategy or adjuvant treatment after surgery. So finally, to summarize, cancer diagnostics and treatments, they improve constantly. There are new treatment strategies and discoveries every year. Genetic alterations are very useful biomarkers for many ways, and targeted treatments have improved prognosis of many cancers and will continue to improve prognosis in future as well. functional imaging and disease monitoring are very important aspects to counter the emergence of resistance as early as possible and adapt treatments accordingly. Currently, I think that the NGS is routine clinical practice, but the next big issue is to understand how the interplay between cancer cells and tumor microenvironment really works, and how we could utilize that to use combinatory treatment strategies and get a full use and benefit out of immuno-oncological treatments. Over the last 12 months, a couple of big things in adult oncology have been the utilization and adaptation of the novel ADC drugs, as well as the treating the MSI high tumors with immuno-oncological treatments. So that summarizes my overview talk on the successes and challenges on precision oncology.
Thank you. Questions for Dr. Kononen? I have one question, and the minister already learned us that budget is always limited. There's never enough money. If you should put your money on one of the things, would you invest in the immunotherapy or in the precision medicine based on DNA abnormalities?
If I would have to pick one of the two, I would invest more on the immunotherapy approaches because they offer us the glimpse of hope of long-term benefits, and in some cases even, I dare not to say the word cure, but it seems like a long-term response is.
Okay, thanks. Thank you. So the last speaker is Jukka Kanerva, a pediatric oncologist from New Children's Hospital in Helsinki. And before he starts his talk on pediatric precision medicine, I will tell you a little story that Dr. Kanerva remembered me this morning. This is just to relax so that you have full attention for him in the last talk. And Dr. Krämer wanted to visit our Princess Máxima Center in March 2020 with a large team of oncologists and nurses. But in March 2020, something else happened in the world, COVID. So a few days before your visit with your large team, we had to cancel it. And instead of that, they went for a team building event to a hotel in Finland, and they all got COVID.
Only half.
So the take-home message is, if you want to prevent having COVID, you better come to the Princess Máxima Center instead of a hotel. But that's completely beside the topic here. Dr. Krämer, you will speak on pediatric precision medicine at the Helsinki University Hospital. The floor is yours.
Yeah. Thank you, Rob, and thank you for the organizers for the invitation to this wonderful meeting. So I will give a grassroots presentation of pediatric oncology and precision medicine in Helsinki Children's Hospital. So what I think is precision medicine, it's many steps actually. It's the clinical diagnosis, then it's tumor markers in blood like NSC for neuroblastoma or AFP in hepatoblastoma. So even before we do imaging and biopsy, we have some idea about what disease we are working with. Then, of course, as a topic of this meeting, molecular profiling in many ways and also functional drug screening are part of this field. But if you think about treatment in pediatric oncology, so most treatment is based on previous clinical studies, and most pediatric first-line treatments are in international clinical studies. And but then in leukemias and in solid tumors also some current treatments are based on molecular profiling, as imatinib in chronic myeloid leukemia. And then mainly then in relapsed and refractory patients, some patients are treated based on drug screening, sensitivity screening, and often that is combined with molecular profiling. And what we routinely do in Helsinki, so that most of our patients are treated in international phase 3 trials. Two examples here, altogether protocol, which includes many countries in Europe. So we stratify this treatment according to many different things. It's clinical risk factors like patient age and white blood cell count. Then it's immune phenotyping and screening for genetic aberrations, and very importantly, response to induction therapy, the first 4 weeks of therapy. And treatment is stratified according to these factors. Another example is brain tumor protocol for medulloblastoma, where we also use clinical risk factors like patient's age and postoperative residual tumor after the tumor resection, and also then cerebrospinal fluid cytology, so if we have cancer cells in the fluid or not. In this medulloblastoma protocol, large-scale molecular profiling is also used for treatment stratification. All these factors are then involved in the treatment decisions. In Helsinki, in leukemias, the typing of leukemia is based on immune phenotype, and then we do targeted FISH studies for known aberrations, then CGH for copy number alterations, and also targeted fusion gene panels for aberrations which we know that have some prognostic importance in leukemias. And very importantly, to then evaluate the treatment response, we search for minimal residual disease markers, which are not really related to the leukemic leukemia itself, but then enable us to very sensitively to find the residual disease. And all the samples, diagnostic samples, are biobanked. Then in extracranial solid tumors, we also do targeted FISH studies based on histopathology and immunohistochemistry, so that when the pathologist has seen the sample, then they continue with FISH studies. We also use fusion gene panels for these patients routinely, and CGH for copy number alterations. Methylation profiling is used in selected cases, mainly in relapsed and refractory cases, and that's not done in Helsinki, so we send the samples to Copenhagen. Then in CNS tumors, it's very much the same line. But methylation profiling is done for all patients in brain tumors in Helsinki, and these fusion gene panels and mutation analysis, they are custom made in our lab for these brain tumors, because the protocols have very clearly defined changes which have to be studied, and also these are biobanked, or all these tumors. And then in relapsed and refractory disease, we try to use international relapse protocols in diseases where we have these available. This is, for example, in leukemias we have those, and in Ewing sarcoma, but in many diseases there's not like a consensus of relapse treatment. And we also take patients to early phase trials in our own clinical trial unit, or send them abroad for trials if we don't have the trials open. And for these patients we do so detailed molecular profiling as possible, and also the in vitro drug screening. And especially in these cases, as we have heard already many times, international collaboration and collaboration between different fields is extremely important. Then I have 3 patient cases with targeted therapy. So this first one is a 13-year-old boy. At the beginning he had an osteosarcoma in right femur, and localized disease was treated with the Uramos protocol and was in remission for a long time, but then 5 years after diagnosis he got a relapse in the left lung, and the whole lung was then resected and the tumor taken away. And we sent samples to the Heidelberg for the INFORM studies for profiling, and then found an NTRK3 fusion and also overexpression in the sample, which is very rare in osteosarcoma. And then we started with larotrectinib monotherapy and had a stable disease like 7 months. We did not have a real target, tumor imaging target, so that we could have followed the response. And there was one metastasis in the other lung which stayed stable during the time. But then there was a new relapse 7 months after larotrectinib was started, and then at that time the NTRK fusion was not anymore there in the sample, so we lost the targets there. Then 2 cases which have a better outcome, at least until this point. This is a 2-year-old boy with pilocytic astrocytoma. So he had seizures in February 2019 and had a bleeding tumor. It had to be evacuated in emergency operation, and the tumor was partially resected. And then there was another resection done then half a year later. He had a hemiparesis, but otherwise was quite okay. And there was no good treatment for this disease, but there was also in this an NTRK fusion in the tumor. So then after the second operation was in the fall of 2019, so then in spring 2020 there was a progression of the tumor, and then we wanted to confirm the progression in another image after 2 months, and when it progressed, then we started larotrectinib treatment, and there was a good response, and still at this point now, it's now 2 years after starting larotrectinib treatment, so the response has stayed good, and he attends daycare normally, has no motor problems at this point. And then a third patient here about ALK inhibition, also a 2-year-old boy with a tumor in thalamic region and also hemiparesis, and was biopsied in 2 years ago, and the diagnosis was a very rare tumor, juvenile xanthogranuloma, and we found an ALK fusion in that tumor. But because there was no knowledge of the treatment results of ALK inhibition in these kind of tumors, we started treatment with histiocytosis treatment with prednisolone and chemotherapy. But already after 2 months of treatment, there was a progression in the tumor, and we changed then to crizotinib monotherapy. And here we can see, so this is a diagnosis that is then after the biopsy, then this is during or after the chemotherapy, and then the third one is then already after 1 month of crizotinib treatment. The tumor had shrinked a lot, and it has stayed this small. It has not disappeared, and this child is also doing fine at the moment. All these 3 treatments have been off-label, so we have not been able to include these patients in trials. And with these 2 patients still on treatment, one difficult question is that how long to treat? Are we courageous enough to stop the treatment at some point? So they have both having treatment already like 2 years or so. So that's all I had to say. Thank you very much.
Questions from the audience, please? No questions. Thank you.
Okay, thank you very much.
And we are closing this first session. I would like to say thank you for all speakers. It was an excellent presentation, and hope we have discussion during our lunchtime. Thank you for participating.